This is a Cambridge IGCSE Biology Paper 4 Theory Extended Paper from June 21. Baker's yeast is a single-celled organism that is classified in the kingdom fungi. Figure 1.1 is a drawing of a section through a yeast cell. State one other kingdom that contains organisms that all have structure A. So structure A over here is the cell wall, and therefore we need to pick the plant kingdom. Table 1.1 shows some cell functions. Complete table 1.1 by naming the cell structure responsible for each cell function and give the letter that identifies each cell structure in figure 1.1. So storage of genes, that is the nucleus. Remember, the nucleus contains DNA. The nucleus is here, look, there's the nucleolus and then the nuclear pores. Do not be tempted to pick C, that's a vacuole, so we need to write G there. Aerobic respiration takes place at the mitochondria. These are the mitochondria, so that's E. Amino acids are assembled to make proteins. That takes place at the ribosomes, which are the small dots up here, H. A student made a drawing of one Escherichia coli bacteria. Figure 1.2 shows the student's drawing. The actual length of the bacterial cell is 2 micrometers. Convert the actual length of the cell to millimeters. To get micrometers into millimeters, you divide by 1,000, so that becomes 2 times 10 to the minus 3 millimeters. State the other information that the student needs in order to calculate the magnification of the drawing. So remember, we use this formula triangle IM. So we also need the image length. Describe the similarities and differences between the structure of the yeast cell and the structure of a bacterial cell. So let's break up our answer. So similarities. So what do they both have in common? Well, they both have the cell membrane, cell wall, cytoplasm, DNA. Differences now. Really lay it out. The bacteria cell has plasmids, nucleoid, a cell wall made from peptidoglycan. Really lay it out. A fungi cell has none of these, and its cell wall is made out of chitin. In addition, the fungi cell has a nucleus and a vacuole and mitochondria, whereas the bacterial cell does not. So we've made a full and complete comparison in terms of similarities and differences between the yeast cell and the bacterial cell. Just going to really show that the fungi cell is the yeast, even though it's already told us higher up. Some bacteria are involved in the nitrogen cycle. Figure 1.3 shows part of the nitrogen cycle. State the processes that are represented by 1, 2, and 3. So nitrogen gas in the air is fixed into ammonium ions, which is why number 1 is nitrogen fixation. What about number 2? We're taking our amino acids, converting them to ammonium ions. This is known as deamination. And then number 3, very important process which adds nitrate ions to the soil, nitrification, which remember is carried out by nitrifying bacteria. Large quantities of plastic waste are polluting the oceans. A survey published in March 2018 showed the increase in plastic waste in the Pacific Ocean. One area of the Pacific Ocean is known as the Great Pacific Garbage Patch, GPGP. Data were collected from areas inside and outside the GPGP between 1965 and 2015 to estimate the quantity of plastic waste. The results are shown. Wow, we've got a big increase in the quantity of plastic waste throughout the years inside the GPGP. It's a bit more variable here outside of the GP increases, and then from 1995 to 2004 it decreases before increasing again. Describe the results of the survey shown in figure 2.1. Hopefully you can see that the quantity of plastic waste is always higher inside the GPGP compared with the light grey outside. We can see here that these inside GPGP numbers are almost constant from 1965 to definitely 1975 and almost 1985. The quantity of plastic waste inside the GPGP increases steeply from 1995 to 2015. And there's a smaller overall increase in plastic waste outside of the GPGP. So we're making 4 different points here. The green turtle is a species of marine animal that is harmed by plastic waste. Figure 2.2 shows a green turtle swimming past a plastic bag in the Pacific Ocean. Turtles are classified as reptiles. State one feature shown by all reptiles that is not found in amphibians. Reptiles have scaly skin. Outline the dangers of non-biodegradable plastic waste to marine animals such as green turtles. Let's first of all say that non-biodegradable waste does not break down using microorganisms, so it persists. If swallowed, it fills the turtle's stomach and stops it feeding. Meaning that you could end up with starvation. Plastic waste may trap the turtle, restricting its movement. And stopping it from escaping predators, so it's more likely to be caught and eaten. The plastic waste may even be toxic. Suggest ways to reduce the quantity of plastic waste so you could seek non-plastic alternatives. You could use biodegradable plastics. You could recycle plastics. All of this will stop this plastic waste being dumped in the oceans. Bacteria are used in many biotechnological processes. Explain why bacteria are useful in biotechnology. Because they reproduce fast. They have very simple requirements. They have plasmids, which remember those small loops of DNA that can actually be used to insert genes into, and therefore those plasmids and the bacteria themselves are acting as vectors. There are no ethical concerns with using them. This is the problem with testing on animals, obviously, and quite rightly people don't like them being killed to test makeup and medicines. Insulin is one of many human proteins that are made by genetically engineered bacteria. Some people cannot produce insulin because their immune system has destroyed the cells that make insulin. State the organ that contains the cells that have been destroyed. It's actually the pancreas. Remember, the pancreas makes insulin. State the name of the disease caused by the destruction of these cells. Ideally, you'd point out that it was type 1 diabetes. Remember, that has a genetic cause. State the function of insulin in the body to lower blood glucose levels. Remember, it converts the glucose to insoluble glycogen. Genetically engineered bacteria that are used to make insulin were grown in a fermenter for 5 days. Samples were taken from the fermenter every 6 hours, and the number of bacteria in 1 millimeters cubed of the nutrient solution were counted. Changes in the numbers of living bacteria in the samples taken from the fermenter are shown in figure 3.1. Complete figure 3.1 by adding labels for the axes. So on this side we want to say population of living bacteria, and that's per millimeter cubed, and then label 2 time in hours. State the names of the stages of population growth of the bacterial labelled P to S. So P is the slow phase, it's the lag phase. Q, we can see exponential growth, huge increase in bacterial populations. R is staying the same, so it's the stationary phase. And then S, massive decrease in numbers, that's the death phase. Explain with reference to figure 3.1 why the bacteria do not grow in the fermenter for longer than 5 days. So what would cause mass death? It could be due to toxins building up, which would kill the bacteria. It could be too high a temperature, so it would denature the enzymes in the bacteria. It could be due to lack of food and oxygen after all those bacteria have been busy respiring, and also overpopulation leads to lack of space. Mineral salts are important in the human diet. One of the most important is iron. Explain the importance of iron in the human diet and the effects of the iron deficiency. So iron is needed to make hemoglobin in red blood cells. So that oxygen can be transported. That's enough about the importance. What about iron deficiency? It's known as anemia, and that means you get very tired, you get fatigue. Lacking in energy. Figure 3.2 shows a field of cassava, which is a crop plant grown in parts of Africa and Asia. The plants store starch in their roots, which form a large part of the diet for many people, but cassava does not provide many vitamins or mineral ions. Genetic engineers have modified cassava to increase its iron content. They've done this by incorporating a gene for a membrane protein from the plant. State the name of the enzyme that is used to cut the gene from the DNA of the plant. Remember, that is a restriction enzyme. Describe how the gene from the plant and the DNA from cassava form recombinant DNA. Use the same restriction enzyme to cut open the DNA of the cassava. Use a ligase enzyme to join the gene to the cassava DNA. Sticky ends seal. Scientists who develop genetically engineered varieties of crop plants often breed them for several generations before releasing them for farmers to use. Suggests why the scientists do this. Firstly, to check that the offspring plants have inherited the new trait. and secondly to build up a large stock of seed. Two identical potted plants were used to investigate plant responses. Plant A was placed on a clinostat that continually rotated. Plant B was not rotated. Both plants were then placed on their sides and kept in the dark. Figure 4.1 shows the two plants at the start of the experiment and after 7 days. So here's our clinostat rotating, and then we're in the dark for both. And then after 7 days, as we can see, plant B has started to bend its stem upwards. State the name of the response shown by the shoot of plant B. Because it's in the dark, it can't be responding to light, it's actually responding to gravity, so it's gravitropism. Explain the reason for constantly rotating plant A. That is, the clinostat's purpose is to negate the effects of gravity. After all, we're investigating gravity here. And this will enable plant A to be used as a control to compare it with the response of plant B. State the name of the plant hormone that causes the response of the shoot of plant B. Those are auxins. Explain how the plant hormone causes the response of plant B. So like I said before, the stem is pointing upwards. How does that get brought about? Well, the auxin diffuses away from the tip. Auxin causes cell elongation. So that's the extension of those cells. And therefore the stem bends upwards. Seeds germinate in the soil. The seedlings that grow from seeds show the same response as shown by plant B in figure 4.1. Explain the advantages of this response to the survival of seedlings and mature plants. So why does the seedling stem respond like this? To enable plants' leaves to absorb more light. After all, they're closer to that sun, which is needed for photosynthesis. The leaves absorb more carbon dioxide, again for photosynthesis. And they can also absorb more oxygen needed for respiration. Figure 5.1 shows the female reproductive system. Label 5.1 using the letters listed to show the position of the organs that are identified by their functions. The first one, P, has been completed for you. So P is the site of secretion of mucus, so that's done. What about Q, the site of fertilization? That takes place in the oviduct. So pick either the left or right side. R is the site of implantation, so that's the lining, the endometrium of the uterus. S is the site of estrogen secretion, which is one of the ovaries. T is the site where sperm are deposited during sexual intercourse, so we need to label the vagina. Figure 5.2 shows a section through an egg cell at the time of ovulation. We have the jelly coats surrounding the cytoplasm that contains stores of protein and fat, as well as the nucleus. Explain why the egg cell contains stores of protein and fat. So the fat is needed to make cell membranes, because after all, we're forming a zygote and then an embryo, so it'll need lots of cell membranes, as well as cytoplasm. The protein is there to make enzymes. And then both are needed as a source of energy. Describe the function of the jelly coat. Well, remember, this hardens once fertilization has taken place in order to prevent more sperm entering. Fertilization results in the formation of a zygote. Describe how an embryo is formed from a zygote. So the zygote divides by mitosis, which removes another type of cell division. To form two cells which are genetically identical. Further mitosis forms 4 cells, 8 cells, 16 cells, so we can see how our embryo is being built up. And then differentiation occurs of the stem cells to form lots of different cell types. Antibodies are proteins that are produced by lymphocytes. Antitoxins are antibodies which neutralize the toxins released by bacteria. The transmissible disease diphtheria is caused by a bacterium that releases a toxin that can cause serious damage to the body. A person is suspected of having caught diphtheria. At a clinic, the person is given an injection of antitoxin antibodies that provide protection against the diphtheria toxin. She's also given an injection of the vaccine. A few weeks later, she's given a second injection of the vaccine. Figure 6.1 shows the changes in concentration of the antitoxin antibodies and the antibodies produced in response to the vaccine. So here we're injecting a huge number of antitoxin antibodies, which will try to neutralize the toxins produced by the diphtheria, and over time we can see them dropping off. And then, as you'd expect, with the injection of the vaccine, the antibodies are slowly increasing as those lymphocytes respond to the antigens inside the vaccine. So they start producing antibodies. There are some memory cells made, and then the second injection of vaccine means that those memory cells can rapidly produce large numbers of antibodies. Explain the advantage of giving the person an injection of antitoxin antibodies. This will provide an immediate and fast protection against the diphtheria toxins, and also notice this is a type of passive immunity. Explain how the 2 injections of the vaccine result in better protection against diphtheria than the injection of antitoxin antibodies. So the vaccine provides active immunity. Remember I said higher up that memory cells are produced which provide long-term protection against the diphtheria. And then the second injection means that antibodies are produced sooner, faster, and in much greater quantity, which reduces the chance of getting the disease. So remember that second part of the graph where the number of antibodies was increasing greatly. Explain how antibodies protect the body against pathogens. It's worth 4 marks. Talk about the fact that the pathogen has protein markers known as antigens, which are complementary in shape to the antibodies. Each antibody is specific to a particular pathogen. And then what does the antibody do? The antibody makes it easier for phagocytosis to take place, which is all about engulfing the pathogen, and helps stick together the pathogen, which if you're feeling really fancy is known as agglutination. Antibodies can travel through the body in the lymphatic system. State 2 functions of the lymph system other than defense against disease. The lymphatic system is needed in the transport of fat and also helps drain tissue fluid.